Hello boys, welcome back to another session of online biology video tutorials, and this session is meant for class 12th biology students, and this is for 3rd July 2021. We have been, as you know, discussing, deliberating on chapter number 2, that is sexual reproduction in flowering plants. And in the last few days, we were talking about what is a flower, why it is considered as a reproductive unit of a plant, how the male reproductive organ, namely stamen, will be there, and what is microsporogenesis, how microspores are formed after meiosis. Then we discussed about female reproductive system, that is pistil or carpel. We also talked about the formation of megaspores under megasporogenesis. I also said not to confuse between PMC, pollen mother cell, and MMC, megaspore mother cell, that give rise to respectively microspores and megaspores. Out of the 4 megaspores, 3 will degenerate, 1 becomes functional megaspore. Then we talked about how the pollen grains from anther should reach the stigma, maybe of the same flower or another flower of the same plant, or third version, xenogamy, quite different flower of another plant. And we call this as pollination. And there are agents that help in pollination. We talked about them: anemophily, hydrophily, entomophily, ornithophily, and chiroptophily. So all these are the various agents through which the pollen grain falls on the stigma. So continuing with that, now that the pollination has taken place, the pollen grains are present on the stigma like this, and there is a synchronization, pollen pistil synchronization is taking place. And then what happens? Once the pollen grain falls on the stigma will produce certain enzymes. Because of the presence of those enzymes, the pollen grain starts germinating and forms pollen tube. So it is a situation where it is tube within the tube. This is style, and within the style you have got pollen tube. So 2 tubes you can see. So the pollen tube will be formed and enters up to synergids of the micropolar region. Okay, synergids of the micropolar region, as you are seeing here. Look at that. And each pollen grain will undergo mitosis, I said, and form 2 nuclei. Though he calls it sperm, I would prefer to call it as male nucleus 1 and male nucleus 2. So there is now a journey for the male nuclei. They start traveling along the pollen tube and reach the egg through synergids. And of course there is, I said, if you recollect from your memory, the filial apparatus, which will help in the movement of the pollen tube through micropolar region. And the male nucleus 1, the moment it enters inside, fuses with the nucleus of the ovum. Again, I don't prefer to call it as egg, I prefer to call it as ovum. That means there are 2 nuclei, male nucleus 1 and nucleus of the ovum. These 2 fuse together and form zygote. And this fusion, we call it as syngamy, or in common language, fertilization 1. So what is fertilization 1 or syngamy? It is the fusion of male nucleus 1 with the nucleus of the ovum to produce zygote. So the formation of zygote is an indication of completion of syngamy. Hence, syngamy is referred to as fertilization 1. So you can see here the formation of this, and once the zygote is formed, what is the fate of another nucleus, that is male nucleus 2? That male nucleus 2 will further move and combine with the polar nuclei, which are 2. So there are 2 polar nuclei. Now the male nucleus 2 fuses with the 2 polar nuclei, forming what is known as endosperm. It is called primary endosperm cell, and later on it develops into pen, primary endosperm nucleus. And the pen starts forming a matured endosperm, which will be nutritive tissue for the growing ovule or for the growing fruit as such. So since there is 2 things that is happening, one is the fertilization 1, and then second is fertilization 2. 2 fertilizations are taking place, so we call this as double fertilization. So double fertilization, I can say, is the fertilization. I can say double fertilization is equal to fertilization 1, which is syngamy, plus fertilization 2. So this fertilization 2 is also known as a triple fusion. I can write it as TF, triple fusion. Why triple fusion? There is fusion of 3 nuclei, male nucleus 2 and 2 polar nuclei. So 2 plus 1, 3, triple fusion. So what is double fertilization? Double fertilization, DF is equal to S plus TF. So you can write it as double fertilization DF is equal to syngamy plus triple fusion STF. So you can remember it easily in the exam. DF is equal to STF. Syngamy plus triple fusion. What is syngamy? Fusion of male nucleus 1 with the nucleus of the ovum to form zygote. Since it is 1 plus 1, it is zygote will be diploid. What is double fertilization 2? It is triple fusion. Why triple fusion? It is the fusion of male gamete or male nucleus 2 that fuses with the 2 polar nuclei. So 1 plus 2, 3, triple. 3 nuclei are fusing, hence triple fusion. In syngamy, zygote is the product. In triple fusion, it is the pen, primary endosperm nucleus. I hope I have made clear. And now the synergids, once the double fertilization takes place, the synergids will degenerate, and we start seeing that the endosperm starts developing. Let us see how this fertilized embryo sac. Now you can see the degenerating antipodals also degenerate, synergids also degenerate. You got a zygote and a pen. Now let's see what happens to this fertilized embryo sac. Now since 2 types of fusion, syngamy plus triple fusion, take place in an embryo sac, it is called double fertilization. It is an event unique to flowering plants. The central cell after triple fusion becomes PC, primary endosperm cell, okay, as you are seeing here, and then it develops into pen, primary endosperm nucleus, and zygote develops into embryo. Now here you can see the fertilized embryo sac will be having all these structures, and once it becomes mature, there is degeneration of synergids as well as antipodal cells. Further, we can say once the zygote is formed, once the fertilization takes place, there are certain events that take place. All these are together is known as post-fertilization events. So post-fertilization events will have some structures. What are those? Basically, we can discuss this under 4 events. One is the endosperm development, Second is the embryo development. Third is ovule formation or ovules becoming matured into seeds. And finally, ovary becoming fruit. So, technically speaking, if someone asks you what is a fruit, as a biology student, you have to say developed ovary. Developed ovary is a fruit. Similarly, what are seeds? Developed ovules are seeds. And further, I must tell you here that double fertilization is a big question, mega question. It's a 5-mark question. So you need to draw the vertical section or longitudinal section of carpel, and you have to indicate the micropylar region, then a 7-celled 8-nucleate condition, integuments you have to draw, then ovarian walls you have to draw, pollen tube you have to draw, style and stigma, all this you need to draw. So keep practicing it because it is 5-mark question. So first you must draw, practice drawing longitudinal section of carpel, and then you discuss about what are the events that take place, and finally you conclude what I said, that DF is equal to STF. Then you will get full marks. And underline all the technical words, like you say syngamy, underline that. Triple fusion, underline that. Double fertilization, underline that. PEN, primary endosperm nuclei, underline that. So that the evaluator understands that you have understood the concept. Now, coming to this endosperm development, you can see polar nuclei and egg cell in one stage, and then the sperm fusing with the cells, zygote formation. Now, the PEN, that is polar nuclei fusing with male nucleus 2, forming endosperm. Now you can see the development of zygote into embryo. Now, first to start with, zygote will be having single cell. Now it starts undergoing mitosis, forms 2 cells. Now sooner there will be a globe-like structure at the bottom. So the name globular embryo will be formed. And then there is formation of heart-shaped structure with the suspensor. And this heart-shaped embryo will lead to formation of a matured embryo, each becoming 2 structures basically. One that goes into the soil and becomes a root in future is called a radicle. One that goes up towards the sky and forms the stem is plumule. And of course, there will be 2 coverings over the seed. These seed coverings are called cotyledons. Depending upon the type of plant, 1 cotyledon may be there, monocot we say, and there will be 2 coverings for the 2 seeds. Then we say if 2 coverings are there for the seed, we say dicot or dicotyledonous plants. Further, we can see endosperm development. You can see the large space that is being occupied by the endosperm. And when this process is going on, at the same time, hand in hand, degenerating of synergids and degeneration of antipodals will be taking place. So you can see here synergid cells, egg cells, central cell, and endosperm starts growing. You can see the region, white region. So primary endosperm cell divides repeatedly to form triploid endosperm tissue. Endosperm cells are filled with reserve food materials. This is the thing that you must remember. They are used for nutrition of the developing embryo. So endosperm is a nutritive tissue that helps in developing embryo. In common endosperm development, PEN undergoes successive nuclear divisions to give free nuclei. This stage is called free nuclear endosperm. So there will be many nuclei being formed and they are all free. So number of free nuclei varies greatly. Endosperm becomes cellular due to cell wall formation. So there will be formation of cell wall. Normally, all of us during summer or sometime when we are thirsty, we drink coconut water. That tender coconut water is free nuclear endosperm. It's made up of thousands of nuclei. And the surrounding white kernel which we consume, which we eat, is the cellular endosperm. So whenever you are drinking coconut water, you must remember that what you are drinking is free nuclear endosperm liquid. And when you are eating that coconut, white, fleshy one, you should remember that it is cellular endosperm. And as we talked about it, embryo develops at micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after the formation of some endosperm because it requires nutrition. This gives nutrition to developing embryo. So in monocots and dicots, seeds differ greatly, but embryogeny, that is development of embryo, is similar. How that? So first it is the zygote formation. Zygote becomes proembryo. Proembryo becomes globular shaped embryo, heart shaped embryo, and finally matured embryo. This is how the embryo development takes place. And it has got axis, okay, direction. It has an embryonal axis with the two cotyledons, as you are seeing, cotyledon 1 and cotyledon 2, two cotyledons. The portion of the embryonal axis above the level of cotyledon is the epicotyle. So you got the above one, epicotyle and hypocotyle. The cylindrical portion below the level of cotyledons is hypocotyle that terminates with radical. So radical is the future root, and root will have root cap for protection. The root tip is covered by root cap for protection. Whereas in monocot, you can see the presence of scutellum and the cotyledon, and you have got a coleoptile, then shoot apex, epiblast, and of course radical and colorhiza, again a special nutritive tissue. So in monocot, they possess only one cotyledon, only entire seed is covered with one cotyledon, one seed cover. Cotyledon of the grass family is called scutellum, okay. It has got a special name, scutellum in the grasses. It is situated lateral to the embryonic axis. Lateral means side. As its lower end, the embryonal axis has the radical and root cap enclosed in colorhiza. Now this is again normally one more question. What is colorhiza? Where do you see this? An undifferentiated sheath present in the monocot seed is colorhiza. The portion of the embryonal axis above the level of attachment of scutellum is epicotyle. So here that epicotyle will be there. It has shoot apex. This is epicotyle and this is shoot apex, and a leaf primordium which is going to form the leaves. It is enclosed in coleoptile. So in this region it is enclosed, coleoptile region, hollow foliar structure. So this is how the development of embryo takes place, and in the next class we will talk about seed formation. Until then, draw